Really excited to have Dan O'Connor here from Johns Hopkins University Neuroscience Program. So Dan got his, in addition to being just a great guy, he did his graduate work at Princeton in neuroscience, neurobiology.
Neuroscience.
And molecular biology, sorry. And went on to do postdoctoral work at Janelia, and that's where at least I became, and a lot of people became familiar with his work that kind of ignited a fire in the Vibrissa community and beyond, where he did fantastic work on active sensing in the rodent Vibrissa system in the mouse. So he's done great work. He moved on to take a faculty position at Hopkins, where he is now an associate professor, so he's promoted and gone through some of the hurdles of academia, and he just does fantastic work that's very sort of simultaneously exciting as well as detailed and rigorous. And so if you read his papers, one I remember in particular from 2010, I think, was a J-neurosci paper with 20 figures in it. I don't think that's common. It's quite massive paper, but he really sort of set the stage for a lot of the behavioral work that's going on in this community in active sensing. And so he's told me he's got a lot of things going on, and what he's going to focus on today is sort of a new direction in motor control. And so he's sort of like taking it for a test run in front of us. So I will turn it over to Dan. I hope you enjoyed the talk. I think you will. Thanks.
Great. Thanks, Kurt. Can everybody hear me? All right, so yeah, for better or for worse, in addition to those 20 figures, there were some supplementary figures. So I don't know if I'd do that again. But yeah, thanks for the invitation to come speak here. So very excited to be here. It's my first visit to Georgia Tech. I've had a great day so far, and I'm looking forward to everything to come. So my lab works on the sense of touch, and we do this at levels of the nervous system ranging from the peripheral mechanoreceptors that transduce mechanical stimuli into action potentials, all the way up to sort of interactions among different regions of the cerebral cortex. So touch is to a large extent an active process. So we're often moving our sensors, say our hands, or if we're a rodent, our whiskers around to seek out new tactile information. And in turn, that tactile information often guides new movements. And so as I say, I work on the sense of touch, but as somebody interested in active touch, it's kind of hard to stay away from movement and motor control and trying to understand that a bit. And so as Garrett mentioned, I'm going to describe a new project in the lab. It's really our first foray into motor control proper, and so your feedback is especially helpful there. And so I'd like to say now also that this is, everything I can tell you is the thesis project of one graduate student in my lab, Manny Zhu. I'll show his picture at the end. All right, so motor control. This is also a daunting audience because many people here know way more about motor control than I do. So, but many movements. Okay. Many movements in the world, right, are sequences of component movements, strung together into some larger sequence. So, for instance, birdsong is a famous example where you have syllables strung together into a whole song. There are many examples, but one from say just everyday sporting life would be if you're a soccer player, right? And imagine that you're a striker and you've just received the ball, and you see the goal in the distance. There's a goalie in the goal, and so what do? You plan after you receive the ball. You start executing a movement sequence to dribble the ball down the field. So each individual dribble requires its own has its own sort of motor control problem, its own kinematics. But so you do a number of those. You get within range, and then maybe you do some maneuver to sort of evade or trick the goalie, and then you're going to kick the ball into the goal. So a number of different sort of component movements strung together in a chain. But of course, not only are these complex sequences often learned, as soccer skills would be, but also they depend in sometimes really dramatic ways on sensory input. So for instance, if you're again, that striker dribbling down the field, but now a defender comes and steals the ball from you. You don't just keep dribbling towards the goal with no ball. Instead, you use your sensory input to detect that the ball has been stolen. Then you do something completely different. You branch that motor program. Perhaps you turn around and go run to chase the defender. So it's this kind of flexible, sensory-guided motor sequences that we're trying to understand for the in the work that I'm about to tell you about. So of course, we're not going to bring soccer players into the lab and try to get at this. Instead, we're going to use a method that will be familiar to many of you here, which is the head-fixed mouse. Okay, so we've developed a new task that I think has many of these properties, and I'm just going to jump right in and start describing the task right away. Okay, it's very important that everybody understand this task. So please ask questions if there's something that's not clear. Okay, so so what we're looking at here is a bottom view here of a head-fixed mouse. Okay, this is a somewhat out of focus side view. And I'm going to play a movie that illustrates one trial of this task. Okay, but I'll describe it quickly first. So here is a metal tube. It's we call this a lick port. It's basically a target. Okay, and so this is a touch-driven motor task for the tongue. So the mouse's task when the trial begins is to there's an auditory cue that tells the mouse the trial has begun, and its job is to stick its tongue out and hit the target at that point. Okay. Now, if and only if it makes a contact there, if it touches it, okay, now that's going to trigger movement of this port to a new location, and then the mouse needs to stick its tongue out and hit that one, okay, and so on through a series of 7 locations until it gets to the other side. And if it's made it all the way over, then it gets a water reward. Okay, so it's self-paced, and it requires the mouse to actually hit the port at each moment. Okay, so here this is. I'm going to play the video. This is slowed down 20-fold. So there's a little startup lick, and then the mouse directs the lick over there. Okay, that rightmost location, we're just going to refer to it as R3. And then there's a second rightmost location, R2, and so forth. This is the middle location here, which we call just mid location. Now it moves on to first left location, second left location, third most, that's the final location. And so now after a delay, you're going to see some water come out of the port. That's its reward for having completed the entire sequence. Okay, so there's the water reward. Now, after a delay, a trial begins, and a new trial begins, and now the mouse has to advance the port back in the other direction. So it goes one way, right to left, and then on the next trial, left to right, and so forth throughout the entire behavioral session. Okay. So as I mentioned, some of these sequences of, you can see this is sort of a complex sequence of movements, right? That each component involves some motor control problem of targeting the tongue to a specific defined location, and they have to be strung together in this coordinated sequence, and then a different sequence going back the other direction. As I mentioned, many of these real-world motor control problems like this depend in a very stark way on sensory input. And so by design, we've built something into the task to get at this. So that's we do this by playing a trick on the mouse. Okay, so which is schematized here with these green arrows. Okay, so in most trials, the mice just go from right to left and then back, as I already illustrated. But on a small fraction of trials, okay, when the mice get to the mid location, what happens is we're going to jump the port back 2 steps, and so the mice need to figure out that it's jumped back by, using their tongue. All this is in the dark, by the way; they can't see this, and then resume the sequence, right? And we do something similar going in the other direction. So we call these backtracking trials. So over here, I'm gonna show what that looks like. This is now slowed down 10 times. Okay, a little startup lick, and then the mouse advances the port here. Now we're at the middle. We backtrack, the mouse thinks it's over there, realizes it's not, picks it up at the end. Okay, so mice get really, really good at this. And so I should say they are trained, they know that this can occur. Right? This is a small fraction of trials, but they know that these trials can occur. So the mice get really, really good at this. So we've been looking at this slowed way down. And over here, I'm going to just play this in real time so you can see just how fast the mice are. And you better watch, because otherwise you're going to miss it. Oh, I'm sorry. Here we go. All right, so they complete the entire sequence of 7 locations in about 1 second. OK. So so here is sort of a schematic depiction of this task. Okay, we've got the port positions, the lick port positions, labeled here, rightmost, secondmost, and so on. These little red arrows indicate the touches that drive the port from one location to the next. These shaded gray areas indicate licks. Okay. Here's the auditory cue, right? So that we begin there, and then the mice advance the port through a series of locations, and then eventually get a water reward and keep licking to sort of lap up the water. All right. So we equip this lick port with a 2-axis force sensor here to estimate the vertical and lateral forces that the tongue is exerting on the lick port, just as a way to quantify some of the sensory input. And out of that, we get sort of traces that look like these. As you saw, we do high-speed video as well. So what we do, the way we process that is we use deep neural networks, one to just score the tongue as inside or outside the mouth. And then for the ones that are outside, okay, we use a regression network to get this vector from the base of the tongue to the tip. It looks like this. Okay, so we are going to extract 2 kinematic variables here of potentially more, but these are the ones that we're working with here. The tongue length, which is just the length of this vector, and we're going to call that L, and the angular deviation of the tongue from the midline, which we'll refer to as theta. All right, so we then get additional time series extracted from the video that look like these. So here's the tongue length, L. You can see basically these series of protractions and retractions. Also the time derivative of the tongue length here. And then, of course, the tongue angle, theta, which, as you can see, sort of progresses systematically over the course of the trial from one side to the other. And you can also see that there's some individual modulations of the tongue within each individual lick cycle. They don't just shoot it out in a straight line. They kind of do a little arc thing like that. All right. So that's the behavior and the kind of behavioral data we're dealing with. And if anybody has any questions about that per se, this is a good time to ask. Okay. All right. So I will just say at the outset that, what, yeah. Yeah.
So there's just be one example here, but it looks like in just the touch ones before the water comes, it looks like there's more kinematic variability. I see a lot more frequency wiggles there, whereas when they're just post-water cues, it looks much smoother. Am I making that up?
So you're saying that
the difference in the exploratory ones when licking just to do the 7 touches versus when it's collecting water?
Yeah, that's actually a good question, and I mean, we've done a lot to sort of quantify the licks that advance the thing through, and the ones to lick up water. We've got the data. I don't really have a nice, concise description of that, but I will definitely, can get back to you on that. Mm-hmm. It's a good question. All right, so on a batch of the exploratory. The way we began was just to, basically begin by taking casting a really wide net and just looking for cortical areas that are involved in this task. Okay, and so to do this, we used an optogenetic screen approach. So we used a clear, so-called clear skull preparation, where the skull's, rendered translucent. So the skull's intact here, but you can see the, the blood vessels below this. And these are mice expressing VGAT-CRE AI32 mice. These are mice that are expressing channel rhodopsin in inhibitory neurons. And so when you illuminate with light, you stimulate the inhibitory neurons, you silence the cortical neurons in this area. Okay, this is now a well sort of validated technique developed in the Svoboda lab some years ago. So we had a sort of hit list of areas to assay, indicated with these little circles here. So first was the anterolateral motor cortex, or ALM here. It's an area, motor area, well known to be involved in directional licking. It's been characterized quite a bit by the Svoboda lab. Also, the body region of primary motor cortex, or M1B. Also, the region of primary somatosensory cortex responsible for sensing tongue and jaw motions, or S1TJ, as well as the barrel field, or the whisker part of S1, and the trunk part of S1. And I'll just tell you right now that the barrel field here, actually, if we silence this, later we record there, it's not involved in this task at all, which to me is actually really fun, because I basically, as Garrett knows, I've basically spent my whole career working in the whisker system. It's the first time barrel cortex has ever been a control area for us. So, yeah. Anyway, I guess you got to spend decades doing this before before you appreciate that. But anyway, so we're going to silence these areas bilaterally during the task, and we want to not just understand the involvement of these different areas, right, but also how they might be involved at different time points during each trial, because right, they're distinct parts of this sequence, right? You got to initiate it, you got to progress through, then you got to lick up the water, you got to terminate it, right? And so we did this by silencing at 3 different time points during the task, okay, illustrated as follows. So here's the auditory cue, right, and then we've got a series of touches as the mice progress through, and over here, comes the water delivery, and then there's some additional licks to lick up the water. Okay, so the first inhibition time point that we're going to use is at the cue onset, okay, and we're going to refer to this as inhibition at the trial initiation. All right, this assays the ability of the mice to just get the whole thing started, whole sequence started. Okay, we also inhibit at the middle position, which is the 4th touch onset. Okay, this has to be in closed loop because, as I say, based on where the mouse is, because the mouse is under control, so it gets to the middle location when it gets there, and when it does, on a small fraction of trials, we're going to silence. And then finally, we also inhibit at the moment that the tongue first touches the water. Okay, and all these inhibitions are 2 seconds long. And remember, I told you earlier, just for reference, that it takes the mice about a second to get through the trial. Okay. So those are our 3 time points. So I want to show you a series of movies that illustrate some of the interesting kind of phenotypes that come out from this, and that are highlight really the interesting stuff. Okay, so first up is ALM. Okay, I'm going to show you another movie. At top here is basically what you've been the same movies that you've been looking at before. Okay, at the bottom now is an axis. Okay, where there's going to be a little cursor which moves through to see where we are in time that matches the movie up here. On this part of the axis here, you've got the angular position of the whisker, theta. Okay, at the top, you've got the optogenetic stimulation waveform that determines when we're actually inhibiting. So you can see here is the cue. Okay, normally I will just say that it takes the mice about half a second after the cue onset to get this whole thing started. So so let's just play this. So the trial's begun. We are inhibiting. Mice normally would have started long ago. It's basically just frozen in place. At the end, okay, now it resumes, picks up where it left off once the inhibition stops. Okay, so this is a typical trial. In this case, the animal, when we're inhibiting ALM, it can't even initiate the sequence. Sometimes they get the tongue out of the mouth, in which case it doesn't really go far enough. Okay, and it also doesn't really go side to side. It just you just get these little wimpy center licks. But many times they can't even start it like this. All right, next up is the tongue jaw region of S1. Okay, and I think if you look at this plot here, you can see already what's going to happen. Okay, so we begin. Mice have no trouble getting the tongue out of the mouth, starting, but it's as if the tongue angles are randomized. They're just licking all over the place. And then we stop the inhibition, and after a moment, there it goes. The mouse picks it up and makes it through. All right, so their tongues they have no problem sticking their tongue out, deviating it from the midline, but it's as if they just, they're not able to target it correctly, or they don't know where it is. Okay. Possibly via some deficit in proprioceptive sensing of where it is. We don't know at this point. All right, so when we inhibit the M1 body region, and I should say M1 body is what I am illustrating here, but we actually see this for a variety of anterior cortical areas. We see something very unusual here. So let's play this one. So the trial starts, the mice advance through. Okay, here's going to be the water. Okay, so normally the mice lick up the water, so the water's gone, and then stop. But here, the mouse just keeps going for quite some time. Right. So when we inhibit this area, the mice actually have a deficit in terminating the ongoing licking sequence. All right. So now these are just 3 example trials. I will just assert that these are typical of what we find from these different areas in the interest of time. I can show you at the end the sort of group data that backs that up. But just to summarize, what we find from these inhibition experiments is that ALM is important. It's going to be important for sequence initiation. I didn't show you this, but also for lengthening and deviating the tongue, for getting it far enough out and deviating it from the midline. Okay, S1 tongue jaw region seems to be important somehow for getting the angular control of the tongue right, because they get the tongue out, they deviate it, but it's just not going to the right place. And then M1 body plus other anterior cortical regions somehow, collectively are important for actually turning off an ongoing sequence. We were quite surprised by that. Okay, so that's really a rough and ready screen of involvement for different areas at different times in the task. But of course, what we want to do is go in and record activity from neurons and try to understand, what it is about these brain areas that are important for controlling this sequence licking task. So to do that, we used silicon probes to go in and make these recordings. So we used 64-channel linear arrays, and we recorded in a number of different brain areas depicted here in stereotactic coordinates. Each point is a recording site. So we recorded in ALM up here, one of the areas we were just looking at. We recorded in the tongue jaw region of M1 in yellow here. Okay, and we also recorded in the tongue jaw region of S1 over here. Okay, so recorded a number of areas, including some that aren't shown here. But for the purposes of this talk, I'm really going to focus on 3 areas that seem to be important here: ALM, S1 tongue jaw, and then in addition, the primary region of motor cortex devoted to tongue jaw movement, M1 TJ. Okay, so let me just show you some example spike raster data. There's a lot going on here. We don't need to go through this in detail. The reason I'm showing this is really to sort of give a flavor of the kind of single unit spiking responses that we see in these areas. So we have example units for ALM in this sort of column over here, for M1 tongue jaw over here, and S1 tongue jaw over here. Now, we're showing 3 example units in the rows, okay, for each area. Unit number 1, number 2, number 3, and so forth for sequences moving right to left and left to right. And everything is aligned, okay, to these same time points as I showed you before. So for this part here, we've aligned to the middle lick in the sequence, that center lick. Okay, so these at the top are licks. And the activity is aligned to that. Okay, and then Over here, we're aligning to the trial initiation, and over here to the last lick that touches the port, so the termination of the sequence. Okay, so the sort of responses we can see, the sort of diversity include responses like this ALM neuron, which you can see when the trial begins, its firing rate goes up, and then it just stays up during the progression through the sequence, and then when the trial's over, it just shuts off, and that's true whether we're going from right to left here or left to right. We see some ALM neurons whose activity increases at the initiation of the trial and then sort of systematically tapers off as the mice progress in one direction, and so forth. Okay, and in one tongue jaw, we see a lot of this sort of cycle-by-cycle modulation, where these modulations correspond to individual lick protrusions, individual lick cycles. Okay. And then in S1 tongue jaw region, we also see some neurons that have that, that cycle-by-cycle modulation, like this point here. But we also see some other response types, like this one, which really seems to be responding at some point in the sequence and not at others. Okay, so that's the kind of data. Oh, and I should say, so these spike rasters here are overlaid with the average, with the peri-event time histograms of the firing rate. Okay, so this is the complete data set depicted as heat maps, right, for all the several hundred neurons that we recorded in these 3 areas. Okay, each row here is an individual unit, and the color scale just shows its normalized firing rate. And again, we've aligned either to the middle, the midpoint of the sequence, or to the initiation, or to the termination. And again, showing sequences for progressions from right to left or left to right over here. Okay, so these are organized in the left column into these different sort of clusters. The clustering method, how we assign that isn't particularly important. You use different methods, you get a very similar picture. The point I want to make here is that 2 points really is that you get neurons that have these, as I showed before, these sort of activity patterns that are specific for different moments in the task, or sort of have a graded firing rate that moves across the whole sequence. And if you consider them all as a population, you can sort of progress, you can sort of tile the entire behavioral sequence. So for instance, neurons up here in cluster 1, they're very active before the trials start. Okay, and as soon as the trial starts, their activity decays. Okay, and that's true whether we're going right to left or left to right, and so forth. We have a neuron here in cluster 6 where the activity really builds up as the mice progress from right to left. Okay, and so on. Okay, so the other point I want to make about this is that if you look closely here, you can see that that many of these response types are present in all of the areas, okay, but to different proportions. So again, considering this sort of cluster 1 response type, well, it's prominent here in ALM, right, but we can also see that in the M1 tongue jaw as well as in S1 tongue jaw. Now, over here in S1 tongue jaw or in M1 tongue jaw, you can see these little stripy patterns. Right, these are the responses that correspond to individual lick cycles. Okay, and they're in both areas. You can also find some of these neurons in ALM, like this one down here. Okay, but they're much rarer. So we got similar response types, but distributed in different proportions across these different areas. Okay, so we've got these large populations of recorded neurons, and now we want to try to understand what it is about this neural activity that could be important for controlling this behavior. And so, first, before we do that, we need to think a little bit about what behavioral variables are important for this motor control problem. For progressing through these sequences and doing the backtracking and so forth. Okay. By the way, I should have confessed this earlier. I'll just confess it now. Although the task by construction is set up to really understand this, branching of the motor program by sensory input, or in this case, lack of sensory input when the mice send their tongue out and it's missing something on the backtracking trials. I really don't have much to say about that today. This is really a key direction that's ongoing. But just say that now so that when we get to the end of the talk, people aren't like, nah. Okay. So what behavioral variables really are important for these for this sort of sequence of tongue movements? Well, certainly the brain needs to control the tongue length. Okay, so again, we call this L. And so what I'm showing at the top here are real behavioral data, real tongue length measurements, averaged across trials in mice. And down here you can see they're aligned again to that mid position. Okay, and so whether we're going from right to left in blue or left to right, you can see this series of protrusions and retractions as the mice stick their tongue out and pull it back for each target location. The brain could instead keep track of the velocity of the tongue. L prime here refers to that. Of course, the brain needs to keep track of the tongue angle, which has to progress systematically from right to left or left to right. And as I pointed out earlier, not only is there that systematic progression in each direction, but there's also some modulation within individual lick cycles. Okay, and now these dashed versions of these curves correspond to the backtracking trials. All right, so if you're thinking of motor control as some people do in terms of a sort of a dynamical system, if you're in the same spot, you're in the same state, right? Then that determines exactly where you're going next, what the trajectory is. Roughly in this task, you can end up in positions here where, in going the two different directions, we're in the middle and things are pretty much the same, at least what we measure, right, in terms of the tongue length and the angle and so forth. And so, the brain probably, to sort of disambiguate that, the brain probably also needs to keep track of which direction it's going. Is it going right to left? Is it left to right? So we're going to refer to that sequence direction as D here. And then finally, the brain probably needs to keep track of some sort of notion of where it is within the trial. How's it progressing through? So we're going to refer to this as tau, sequence progress. Right? You gotta know, okay, when to start, when to terminate, where you are within the progression to be able to get that, decide on the tongue angle and so on. Okay, so these are behavioral. There could be others, but these are behavioral variables that that we think are important, and actually, it turns out they're also quite orthogonal to one another. And what we're going to do now to assay the potential involvement of these areas in this task is we're going to try to decode these behavioral variables, right, from each of these brain areas on a single trial basis. Okay, from the neurons that we simultaneously record. Okay? So typically we record at once about 30 neurons or so, and we're going to do this using almost the simplest model that we could to begin with, right? Just a simple linear model where we predict the each behavioral variable, tongue angle, tongue length, and so forth, just as a weighted combination of the firing rates of each of the simultaneously recorded neurons, and we do this after smoothing them with a sort of 15 millisecond time scale. So rather fast, sort of a rather fast sort of fine resolution decoding of the instantaneous tongue length, velocity, angle, and so forth. All right, so we're going to do this from each of these three areas, okay? And so you'll see the decoded traces in these different columns for the three areas. So first, let's consider tongue length. Let's try to decode that. So if we decode that from ALM, oh, and I should say the more these, the problem we're trying to solve here is to estimate each of these variables. So L hat, right? We're trying to estimate L here. And so the closer these decoded traces look like to the real ones, right, the better the decoding. So if we decode from ALM, we get some sort of ripples that correspond to these protrusions and retractions, but overall, relatively poor decoding compared with M1 tongue jaw and S1 tongue jaw, where we get nice large cycle by cycle modulations. Okay, and similar for tongue velocity here. Now, if we try to decode tongue angle, theta, we see something that we think is pretty interesting. So when we do that from M1 tongue jaw or S1 tongue jaw, okay, you can see the systematic, progression of the angle, estimated angle from right to left and left to right, and you also see, these sort of modulations that correspond to the, the kinematics of the tongue on individual lick cycles. Okay. From ALM, we see something quite different. So the decoding here is also quite good. So we get, we estimate the angle of the tongue moving as it progresses right to left and left to right and so on. But it's smooth. You see, there are not the little ripples, okay, that accompany the decoded traces from M1 and S1. And so, the thought here is that S1 and M1 are able to better represent the instantaneous tongue kinematics, whereas this sort of smooth representation would be consistent with, say, with a continuous representation of the target angle. Now, if we try to decode sequence direction and sequence progress, what we see is that in ALM we get relatively good decoding. So the point is that these curves are relatively far apart. We're able to tell whether we're going right to left or left to right relatively well from ALM, somewhat less well in M1 and less well further in S1, right? And then similarly for sequence progress, ALM, decoding from ALM, we do a relatively good job. So this looks more like this, more along the diagonal, okay? And somewhat less good in M1 and less good further in S1. And so I think the sort of the picture that emerges from these analyses is, as I said earlier, that S1 and M1 seem to be involved in the online kind of instantaneous control of tongue kinematics, the progression of the angle systematic from one side to the other, as well as the modulations within each individual lick cycle, whereas ALM is sort of consistent with what you might refer to as a sort of higher level or premotor sort of region, seems to have a sort of continuous, more abstracted representation of the target angle, if you want to call that abstracted, as well as the direction of the sequence, right to left, left to right, as well as a better representation of where in the sequence the brain is. Okay. Yeah.
Are these recordings from one hemisphere or both hemispheres?
Yeah, so that is a good question. So for the most part, this is recordings in the left hemisphere, so the decoding, I don't really have that to show you here, but you kind of do a better job decoding, say, in ALM, for the sequences in one direction, but you can decode in both directions. Yeah, that's a good point. Okay, so this measure of sequence progress in ALM, before I had that in quotation marks, what does that actually mean? And so that, I think, is a very unclear question. Like, how does the brain, to the brain, like, what does sequence progress mean? And there are different possibilities, okay? So again, here's our sort of schematic of the task. Well, maybe the brain, or ALM at least, thinks that the motor sequence is over when the mouse has arrived at that final lick port progression. Okay, maybe it thinks it's over when the tongue hits the water and they get the water reward. Or maybe they think that the sequence is over at the moment that they finish drinking the water and just stop and begin the intertrial interval. So I think we get some insight into how the brain is treating this by looking at some example ALM neurons. So here are two neurons, neuron 1 and neuron 2. Again, spike rasters, peri-event time histograms here. Everything now is aligned to that first lick that touches the water. Okay, so here are the licks up top, and the blue here just depicts the time of the water delivery. And so what you can see for neuron 1 is that the moment the water comes, okay, the firing rate of that neuron shuts off very rapidly. And for neuron 2, it's a little more gradual, but something similar. Firing rate shuts off when the water comes. And this is true, for example, for sequences moving right to left. But you can also see here that it's true for sequences in the other direction, left to right. Again, it just shuts off abruptly when the water touches the tongue. All right, so that suggests that that it's the the reward delivery that, from ALM's perspective, from the brain's perspective, terminates the sequence. Here's one of those same set of example neurons, just for reference. Now you might say, okay, well, this is also the licking kind of peters out, and they're not licking as much immediately after the water is delivered. That's not exactly true. So if we look just at the probability of lick from these behavioral data here, and we align and we just count and we just name the licks from the one that touches the water, where that one is 0, the one after is 1, the one before it is -1, and so forth. What you can see is that yes, the licking tapers off, but it persists for a few licks, several licks beyond the time of the water reward, and doesn't really account for the drop-off of the neural activity. Similarly, for tongue length, right? They're continuing to stick the tongue out well after that, the tongue touching the water lick at 0 velocity, angular position, and you can say, well, the tongue isn't pushing as hard against the lick port, but if we look at the sort of total force there, you can see that if anything, that actually grows, after the water is delivered. So it's not some simple response to stopping licking or changing the sensory input, as far as we can tell. And interestingly, if we try to, continue decoding sequence progress, or tau, based on a model fitted to the actual, period during the earlier trial, you can see that the decoding quality really, or the decoding actually sort of drops off shortly after this first lick, suggestive that ALM at that point, considers progress to be resetting. So, and also the somewhat more gradually, the sort of this memory or this encoding of which direction the sequence is moving also comes together, consistent with ALM thinking the sequence is over. So with tongue angle, it's definitely more gradual here. Not exactly sure what to make of that, but it does contrast, for instance, with what you see in, for instance, S1 tongue jaw, where for well after that reward delivery lick, the tongue angle, including its cycle-by-cycle modulations, is still decoded quite well. Okay, so we think ALM is encoding the termination of a sequence as measured by the reward that marks the successful completion of a sequence. So for putting this together into some synthesis here, we think that ALM is involved in initiating these lick sequences. Right? If you silence it, the mice have trouble getting started. Even when they do get started, I didn't show you an example of this, but even when they do, they have a deficit with lengthening the tongue and deviating it from the midline. It encodes some abstract target position, the sequence direction, and some sort of measure of distance to the reward. And I also didn't show you this, but we also find consistent with what you might predict from work, for instance, from the Svoboda lab, that ALM also encodes the target direction on the next trial, because they have to go back and forth. Remember, they alternate, and when the mouse is just sitting there in the intertrial interval, ALM will tell you which side is next, even when the body is in the exact same position. All right. M1 tongue jaw, this seems to encode the instantaneous tongue length and angle, the instantaneous tongue kinematics. And I didn't show you this, but also consistent with work from Carl Peterson's lab recently, we find that if we activate that area, that it does drive tongue jaw movements. This area is actually recently kind of characterized by Carl's lab. The S1 tongue jaw region seems to be involved in proper control of tongue angle, because when we silenced it, the tongue was just kind of going all over the place. And this is speculation at this point, but we think that may be due to sort of loss of proprioceptive feedback, which tells the brain, where is the tongue. And it also does encode the instantaneous tongue length and angle. And then something we're still trying to make sense of is that a number of anterior cortical regions, I showed you M1 body silencing, but a number of anterior cortical regions all seem to be such that you need activity there to terminate some ongoing sequence. So this is sort of just, as I said, a very exploratory beginning to try to understand these tongue motor control sequences. So that's what I have to tell you for today. Now, very important, as I said, this is all the thesis project work of Manny Zhu, super talented PhD student finishing up in the lab. I will also, for the graduate students here, I also say that that this whole thing was his second. I'm quite happy with with how it's turned out so far, and this is all his second project. The first one he had really met with a. It was just one of these cases in science where you do everything right and the biology just doesn't cooperate. So so he did this as his second one, which I think really illustrates incredible persistence with help from the people listed here. So that I can take any questions.
Time for some questions?
The perturbation experiments I thought were really cool. Did you do any unilateral perturbations to see if they manipulate specific variables?
Yeah, that's a great question. We have actually not done those. I think we should do that. We started to at the beginning and got a little bit, realized that was going to be a little more complicated than we initially thought because, interestingly, the tongue being a midline structure, that although it's represented, the two parts are represented in different hemispheres. If you've got, say, some muscles tugging on it, and you interfere with one side and not the other, it's not clear what the net result would be that you'd expect to see, right? The tongue can, and in fact what we found is sometimes that the tongue can sort of change shape, and so we need to come back to this, but we haven't done that yet.
Question on the coding side that you were doing. So you had started with having a set of outputs that you were looking at, and then the set of recordings that you had. And you said you started with the linear model, which is a very good place to start. Were you doing them simultaneously, or were you doing different linear models for each of the different outputs?
So we fitted one model to each of those 5 behavioral variables. A couple points about that that I didn't go into is that each of them seems to be those variables actually are it's maybe not obvious, but they are orthogonal, just the behavioral variables themselves. Okay, and they seem to be, you fit these 5 models, but they seem to be encoded in different neural subspaces. And interestingly, you could say with a situation like this, you can say, okay, you do a great job predicting, say tongue length, but is that really due to the responses of 2 neurons, or does that really capture a lot of what the population does? And in this case, for all these variables, the answer is actually is the latter. So if you do some unsupervised measures of trying to explain the overall population dynamics, like principal component analysis, and you compare that to what we get, the sort of the directions in this neural space that we get from fitting these linear models, and you kind of rotate them, they actually match quite well. So these models actually capture the overall dominant activity patterns in the entire population.
You showed the rasters. You showed there's a big diversity of response types in ALM. They sort of clustered together. I assume for the decoding, you just took like a sort of subset of things. So I was wondering if you're seeing different aspects of ALM, like tribal identity and progression, map onto those clusters of cell types.
Yeah, I mean, well, so we actually, I mean, what goes into the model are basically all of the simultaneously recorded units in each case. And yeah, I mean, you certainly, you know, in some cases, you know, as I was showing with ALM example units responding to determination of reward, you know, you certainly can go in and find, you know, individual units where you see, well, you have, you know, a set of these things, maybe that, you know, I mean, in that case, it's just the termination, right? But, okay, you know, one unit responds to the onset of the trial, another responds to the offset, and you can, you know, you definitely can see how the population, you know, decoding arises from those individual units. I'm not sure if that answers your question.
Yeah, I guess like this topic is it's like an easy example to understand. This stuff like tracking progression
Yeah.
may not be as obvious in just like the rates. So is that map onto like a cell type or depth in the cortex?
Yeah. I mean, right. Yeah. So that is. So we were actually quite surprised. Okay, so, you know, we, you know, absolutely want to kind of, I mean, this is basically a bag of neurons approach that I showed you, you know, and absolutely we want to try to understand this in terms of, you know, what are the cell types, you know, eventually the connections and so on. And for, you know, like I said, this is entirely, you know, one PhD student's project, and we really just haven't gotten there yet. So we recorded with the laminar arrays, so we can at least consider things as a function of cortical depth with some confidence. And you know, we were really surprised, actually, or maybe I don't know, maybe other people won't be surprised. I was surprised that there really isn't a depth dependence to the different, you know, functional types, different clusters in any of the areas. Like to the extent that it's like, check your code, but. Yeah, there really isn't.
I had a question. I think you gave a disclaimer somewhere in the middle that the sensory part of this isn't quite worked out. It'll be really interesting when you close the loop on that to try to understand, especially in those tricks, when you're tricking the mouse with the backtrack. I'm just curious, do you see, and you may have said this if I might have missed it, during those trials where you're tricking and doing the backtrack, are there signatures in the decoding, for example, that are representative of that kind of error?
Yeah. Mm-hmm.
Because, you know, you can kind of imagine the motor planning is kind of clicking along and thinking ahead.
Yeah.
And this correction comes along from somewhere, the
Yeah.
sensory pathway, and says, no, wait, you've got to change your plan based on that.
Yeah. So, I mean, okay, well, yeah, so I don't have a good answer for that. Like, can we, but we are kind of systematically looking, as the next PhD student's project, we are systematically looking to understand, you know, where that error signal comes from, because it's also a somewhat interesting, modestly interesting case in the sense that it's the lack of a tactile input, right, that needs to get converted into some signal which dramatically kind of bifurcates the unfolding motor program.
There's an expectation somewhere, anticipated response.
Exactly. Yeah, I mean, one thing, you saw it in the videos, but you know, one thing that's worth mentioning is that the whole thing is very fast and operates on a, you know, pretty characteristic time scale of 1 interlick interval. And the mice actually, they don't miss a beat. So when they, you know, they're looking for the port, if they hit it, then they move on to the next one. It's a regular trial. You know, they're looking for the port, they stick their tongue out, it's missing. There's no change in the interlick interval. They immediately go back. So that whole computation has to be constrained within the, you know, the period that you get with a 6 hertz or so lick cycle. Yeah.
That's very cool. Any other thoughts or questions? All right, so let's thank Dan again.